pleasure, I have to say. It's my first chance to be a part of the Frank J. DeSantis Investigator Award lecture, and so we're really pleased to be with you here today to welcome our honoree this year, Dr. Ashraf Ibrahim, who was recognized also at this year's recent Legends event. And so this presentation is actually a part of the award. So there's a recognition at the Legends event, and then we want to make sure the community gets a chance to hear about some of the work that's represented by that honor. Doctor, and I know you guys know a lot of this, but I'm going to do it the way I think is best to do this sort of thing. So Dr. Ashraf Ibrahim has been, I think, the very embodiment of the intent of what this award celebrates. He's been an outstanding scientist, mentor, and leader whose pioneering research in infectious diseases has transformed our understanding of fungal pathogenesis and created new immunotherapies to treat the infections that they cause. Dr. Ibrahim continues to lead groundbreaking studies in many different areas. He was just telling me a few new ones, actually, to improve our ability to fight fungal infections that are on the rise and often in the news these days. I wouldn't be surprised if you picked up the newspaper today if there wasn't something in there about some new challenge. This includes his work on mucormycosis, and his research is accelerating vaccine and antibiotic development against multi-drug resistant microorganisms, and it's formed the foundation of several of his companies to try and get these drugs to the people who need them. His impact is also seen in his more than 200 peer-reviewed publications, and he has been supported by close to $100 million in research funding over his career here. His impact extends far beyond our campus. He's been recently awarded the prestigious 2024 Kuwait Prize in Biological Sciences. This was to recognize his pioneering work in translational medicine and the development of innovative diagnostic and therapeutic approaches to complex diseases. More recently, his company Vitalex has just been received $40 million. $40 million is pretty darn good, though. $40 million NIH competitive contract for strategic advancement of second-generation fungal vaccine through the phase 1 trials, which I'm really eager to see that work. He's an elected fellow of the American Academy of Microbiology, a former member of TLI's board of directors, and again, a dedicated mentor and visionary entrepreneur. He continues to push the boundaries of translational science while inspiring the next generation, some of whom are in the room, of investigators. His lecture today, as you can see, is entitled Immunotherapies Targeting Virulent Traits of Invasive Fungal Infections. And so please join me in congratulating him and welcoming him. Thank you, Ash.
Well, thank you, Joe, for your kind words. And it's really a real pleasure to be with you guys. So I'm going to talk to you today about immunotherapies of fungal infections, and I apologize for the misspelling. Triers, it's actually treats, so it's of invasive fungal infections. So let's see if I can get the pointer here to work. Sorry. Well, can you see my cursor? I think you can, right? Perfect. All right, so before we start, let me see, why is this not moving? All right, here you go. My disclosures, as you all know, that Vitalex Biosciences is a spinoff from the Lankus Institute. I'm a founder and the CEO, and this is really the most important one that you need to know about. We do a lot of other work with anti-fungal drug companies as well. Just let me see here. Here you go. So before we start about telling you what the targets are, I thought maybe perhaps we should talk about why are we interested in immunotherapies. And the most important 4 points that I would like to discuss today is over the last 4 or 5 decades, we've had literally no improvement of mortality rates in fungal infections. You know, we still basically get anywhere between 40, 50% to 100%. And it seems like despite the fact that we are basically making very good advances in discovery of antifungal drug agents, we used to have only polyenes, which are very toxic, but now we have several azoles, which are actually have very good broad spectrum antibiotics activity, as well as actually, in static in vitro activity, but it seems to be actually working really well in vivo and animal models. And also we have in the pipeline a lot of other candidates, in addition to the fact that we have echinocandins that have been approved since 2002. We have echinocandin-like antifungals as well. And we have drugs that are actually targeting expression of cell wall proteins, GPI-anchored proteins. They are called phosphomanogepix. We still have that 40 to 100% mortality rate. So it doesn't seem like discovery of new small molecules as antifungal agents is really helping that patient population. Secondly, we are having more and more of a drug resistance, similar to what the bacterial environment is basically facing with with drug resistance. That's basically in gram negatives as well as in MRSAs. We're also seeing a lot of drug resistance in fungal infections, be it basically developed resistance or inherent resistance. For example, azoles, Aspergillus, for instance, is developing actually a lot of azole resistance. We have also, in addition to that, Mucorales fungi. They are inherent to echinocandin effect. They have some elevated resistance to polyenes as well. And then in addition to this, we have also what we call Candida auris, which I'm going to talk about also, that's developing really pan-resistant in certain cases as well to all polyenes, azoles, and echinocandins, which are really the drugs used for treating Candida. Patients are immunosuppressed, so it makes a lot of sense if they are immunosuppressed that you basically boost their immune system. It's simple. If you lack something and that something is basically causing you to be more susceptible to infection, it makes a lot of sense that you try to compensate for that deficiency. And lastly, immunotherapies work really well. So vaccines and antibodies are really proven to be safe. Vaccines was the major way of fighting infectious disease for more than 150 to 200 years. And they were working well until basically the pharmaceutical industry decided to shy away from it in, I would say, 60s, 70s, because of the fact that they were not as profitable as discovery of antibiotics and antifungals as well. So it makes a lot of sense for us to basically continue with the vaccines, because vaccines and immunotherapies are proven to act really well. And it's basically the only method that eradicated an infectious disease, smallpox, and came close to do the same thing with polio as well. So with that being said, I would like to go over what I'm going to cover today. So I'm going to talk about vaccine development against Candida infections, both disseminated as well as mucosal infection. And we're going to target two basically cell wall proteins that are involved in adhesion invasion and host evasion as well. So, and also I'm going to talk about factors of angio invasion and necrosis, and a special fungal infection called mucormycosis. It's a rare infection, but it's actually increasing in numbers, and it's highly lethal. And we're going to talk about a humanized antibody that we developed recently, targeting one of those effectors, which is basically an invasion. And I'm going to touch upon also tissue necrosis by a toxin, but I'm not going to have a lot of time to cover that toxin in details. So we'll see what we get to. So before we get into the actual technologies that we are covering, let me just cover a little bit about the disease itself. So hemidionously disseminated candidiasis is considered to be the third or the fourth most common bloodstream infection in hospitalized patients, depending on what reference you follow and what year you are basically looking at. But the bottom line is there is probably approximately 30 to 40,000 cases in the US per year, and the mortality rate of those hematogenous disseminated candidiasis still hovers around between 30 to 40%. And ever since I was a postdoc, I hear the same mortality rate. Never changes. It's the same. So in addition to this, Candida and majority of the organisms that cause hematogenous disseminated candidiasis are actually Candida albicans. We're still having higher cases of Candida glabrata and other species as well. But Candida albicans is the main problem with his hematogenous disseminated candidiasis. Candida albicans also can cause mucosal infection. There are two major mucosal infections. These are basically usually oropharyngeal or oral thrush in preterm babies and also in HIV patients. And in addition to this, we have, of course, the yeast infection in women. So almost every single woman goes at least of one yeast infection in a lifetime. And among those, there are anywhere between 6 to 9% of women who suffer from what we call recurrent vulvovaginal candidiasis, defined as being 3 episodes or more of yeast infection per year. And of course, it's not lethal, but it comes with a very high morbidity and a lot of basically toll, economical toll on countries. And this is specifically more common in the Western world than anything else. And it usually happens among women who are less than 40 years old. So we're talking about the most productive period of an age. So who gets this infection in addition to these manifestations that I said? You have basically the classic neutropenic patients, weakened immune system with steroids and others. But in addition to this, we have people who have attached medical devices, central venous catheters come to mind. There's also people who go into intra-abdominal surgeries. There's those who are with diabetics and those who are basically in the ICU for a long period of time. So if you look at the last categories, other than the neutropenic patients, these are really not classically immunosuppressed. They do have some immune system, and a vaccine approach hopefully would basically help them to basically develop some sort of a resistance to the infection, especially if the protection is not really needed for a long period of time. A lot of those patients basically have a predefined conditions that can make them predisposed to the infection. And if you can identify them, you do have the time to basically vaccinate them, and hopefully by then you can protect them from infection. So when I came into the Lancaster Institute, used to be actually called Research and Institute, REI, Research and Education Institute. Jack Edwards and Scott Filler. Scott was a postdoc at the time. In his last year as a postdoc, I was coming in my first year. They were working on basically trying to understand how the initial steps of infection happens in Candida albicans. And they came up with a very nice model whereby Candida that is introduced in central venous catheters, basically what they do is once in the blood, it starts germinating. And we know this very well, that if you put some serum, it's one of the actually more potent effectors of germination. It adheres really avidly to the host cells. In this case, as you can see here, these are endothelial cells lining blood vessels. They basically induce their own phagocytosis or endocytosis. They kill the cells and they basically disseminate into the tissue parenchyma. So trying to interfere in early stages here where they basically adhere and invade, hopefully you'll prevent basically further damage to the host. One of the things that actually was always astonishing to me, if you look at the ability of Candida species to adhere to plastic or host tissues, you look at Candida albicans, it's extremely adhesive. You put those things on host cells or on plastic, and within literally 10 minutes, Candida can adhere really avidly. You can actually wash really vigorously, and you can't really get rid of those bugs. I mean, nothing against bacteria, but a lot of people, like when I read the bacterial literature, they talk, and sorry, Mike, but I love Candida because how sticky it is. You know, you look at MRSA and how it basically adheres, and it's nothing compared to Candida. It's not even 1/10 of what Candida can do in terms of basically adhering to host cells and plastic. And then you look at the other species, it becomes less and less and less. And look at Saccharomyces cerevisiae, which is the baker's yeast, has absolutely no adherence whatsoever to endothelial cells. So, and you look at the ability of Candida to cause disease in animals or in humans. We talked earlier and we said Candida basically adheres really well, but it causes the most common causes of bloodstream infection. And you can see in the animal model, if you put some Candida albicans in a mouse model, they drop dead right away, you know, within a few days. Immunocompetent, they are not immunosuppressed. You can't do the same with the other species. you really have to immunosuppress them very, very harshly. So adhesion seems to actually be one of the most determinant virulence factor for Candida albicans. And in addition to adhesion, it's invasion, obviously. If you adhere very well, you can invade very well. So Yu Fu, who joined the lab pretty much within the same time period as me, is a molecular biologist. He basically took advantage of the fact that there is really a huge differentiation in the ability of adhering to host tissues between Candida albicans and Saccharomyces cerevisiae. So what he did is he took basically Candida albicans genome, mechanically sheared it, and then he ligated it into a plasmid under the galactose promoter. And he transformed Saccharomyces cerevisiae with it, grew it on galactose, and we started basically screening for clones that can adhere to endothelial cells. And we found one of the clones which adhered really well when you grow it in galactose, not when you grow it in glucose. This is really a good confirmatory step. And actually, even it adhered much better than what Candida albicans does under the same conditions. So we retrieved the fragment and we identified what we called agglutinin-like sequence protein, or ALS protein. So ALS protein are basically predicted to be cell surface proteins. They have the classic features of cell surface proteins and a signatory sequence, as well as an N-terminus, a very highly glycosylated tandem repeats, and also a GPI anchored protein. ALS proteins basically constitutes 8 or 9 members. They are present in all strains that we've sequenced at the time, 20 of those, blood, urine, or OPC. And also some of those sequences are present in other Candida species as well, including Candida auris as well. And I'll talk about that. So Scott, Filler, and Trang, they basically determined that in addition to the fact that ALS3 is an adhesin, is actually also an invasion. And at the time, in 2000, I don't know, 4 or 5, this was the first invasion to be identified in fungal pathogens. So it was a major discovery. And what they found is actually it adheres to N-cadherin on endothelial cells and E-cadherin on epithelial cells. And you can see here that if you basically look at knockouts of ALS3, you really don't see any co-localization between N-cadherin and the hyphae. So you can see it here in the wild type really nicely with the yellow color. So we basically were buoyed by the fact that this is a very nice, virulence factor. We basically took ALS3 and terminus, expressed it in Saccharomyces cerevisiae, and then we teamed it up with alum, and we used it as a vaccine. This is in early 2000, and in 2006, we formed Novodyne Therapeutics, Inc. And the reason we used alum, because alum was the only adjuvant approved to be used in humans. So there was no other adjuvant, believe it or not. And you can see here, if you vaccinate mice with NDV3A, which is the N-terminus of ALS3, you protect mice very well, approximately 40%. Again, it might not look really very impressive to you, but this is really in a very, very highly lethal model. You can see that within 7 days, all mice that are unvaccinated dropped at 100% mortality. We also determined that that the vaccine required interferon gamma. If you do the vaccination in interferon gamma and knock out mice, you completely lose that protective effect. We also established a mouse VVC model, and we determined that the vaccine is protective, as you can see here, approximately reduction in CFUs by 1 log in mice that have been vaccinated. And it seems that also the vaccine required both T cells and B cells, because if you vaccinate knockout mice of T cells or B cells, you lose that protective activity again. So we've also done 2 phase 1. So we've done a lot of manufacturing, and then we basically went into 2 phase 1 clinical trials and showed that the vaccine was highly immunogenic and extremely safe, no major issues. And that basically prompted Novodyne Therapeutics, Inc. to go all the way into a phase 2 clinical trial in a phase 2 randomized, double-blind, placebo-controlled, whereby we vaccinated, if I remember correctly, approximately 80 maybe patients with NDV3A versus non-vaccinated, and we used only a single dose. And what we found is these are the results. So we'll see that basically in the vaccinated women, 42% of them were recurrent-free after 1 year of follow-up versus 22 in the placebo. Definitely there was, and this is in women who are less than 40 years old, which represents approximately 80% of the patient population of the clinical trial. So clearly there was actually a very significant protection. Wasn't wow, wasn't what we wanted to see, 70 and above or 80% protected, but it is a very good signal. And not only that, but also if you look at the patients who relapsed, received the vaccine and relapsed, the median time for first relapse is basically 200 days versus 100. So the argument here is, had we basically gone and given a second dose, a booster, probably those patients would have been protected for a longer period of time, perhaps maybe for a year. So that was the whole idea. We tried to basically, in Novozymes, to raise funds to basically conduct other clinical trials, but the company failed. There was really no interest at the time in the disease, RBDC, but there was an interest in disseminated candidiasis, and there wasn't really enough money to go and conduct a clinical trial that cost tens of millions of dollars on disseminated candidiasis. So we went back to the drawing board, and we said, we're not going to give up. We're going to basically try to enhance this vaccine. Remember, we said that this is alum adjuvanted vaccine. By the time Novozymes went belly up in 2021, there were many other adjuvants, much better adjuvants, especially those which can induce a balanced immune response between Th1, Th2, and Th17. And they have either shown in clinical trials to be safe or are FDA approved to be used. And that was the whole idea. We wanted to basically use another adjuvant, but also in addition to ALS3, we thought probably we should include another antigen that, at least in animal models, showed even better protection in our hands than ALS3, but we discovered it later. And hopefully we basically also tested against different organisms as well. So around the same time, Candida auris became a serious problem. Although it was discovered approximately 15 years ago as an ear infection in one patient in Japan, it had spread basically in the Western world and in India and in Africa, everywhere, like fire. So we've seen basically major outbreaks on the East Coast in New York, but it kept basically spreading all the way to Chicago, and now we know that it's basically present both in Orange County and LA County as well. Candida auris is a major problem. First of all, mortality rate can be up to 60 and 70%, simply because Candida auris is extremely drug resistant. What I call is a professional drug resistant. Most isolates that are basically isolated these days are completely resistant to azoles, 95%, more than 95%. A good chunk of them are actually also pollene resistant, which is usually very rare in fungi to find basically an organism that is basically resistant to amphotericin B. And then approximately, luckily, 7% are still, majority of them are still susceptible to the first-line therapy, which is echinocandin therapy. So we approximately have 7%, and still, till this day, is around 7% to echinocandins. But you can imagine if you basically have a pan-resistant isolate, you really have no way of treating those patients, at least not right now. Hopefully soon we'll have other small molecules as well. Phosphomanogepix in particular seems to be working really well in animal models. So the second antigen that we were basically looking at is HYR1, which is HIFI regulated protein. This is a protein that is expressed exclusively on the hyphae of Candida albicans, which is the virulent form of Candida albicans. And HYR1, what it does, it basically enables the organism to evade host phagocytes. If you knock out HYR1 in Candida albicans, you increase the susceptibility of Candida albicans to neutrophil and macrophage killing. So we're hoping by basically raising immunity against this one, we would basically enable the host to recognize the bug and eliminate it, in addition to also prevent it from adhesion and invasion. So both proteins, they have orthologs in Candida auris, and they have also orthologs in other species, but I'm focusing on Candida auris because of the tremendous need for Candida auris novel treatment. So you can see here, for instance, ALS3 in Candida albicans looks pretty much the same as at least 3 orthologs expressed on the cell surface of Candida auris. And if you vaccinate mice with NDV3A, which is adjuvanted with alum, you actually protect them very well against Candida auris. And this is in a neutropenic mouse model. And you also look at HYR1. There's more or less the same structure. And if you raise antibodies against a conserved peptide in HYR1, both in HYR1 in Candida albicans or its ortholog, you actually protect mice from infection with Candida auris if you give the antibody approximately 24 hours post-infection. So these are excellent results for us because it means that hopefully the vaccine approach will work. So what we're trying to do here is combining 2 antigens that can target Candida albicans and Candida auris and pair them with adjuvants that are 2nd generation or 3rd generation, if you want to call it, that are proven to be safe or approved by the FDA for human use. And hopefully, by doing so, we'll basically generate a balanced immune response, Th1, Th2, and Th17. We know that most fungal infections require at least a Th1 immune response, if not also some of Th17 as well. And by doing so, we'll target Candida albicans, Candida auris, and other species. And I'm not going to go into another thing, which is gram-negatives, because HYR1 seems to actually have structural homology with hemagglutinin as well on some of the gram-negatives. But this is a story for a different day. These are the adjuvants that we've tested. There are all in all are 7. I'm not going to go through all of them, but they are all shown to be at least to be safe in clinical trials. And by doing a checkerboard approach, we've done a massive screening experiments testing 4 doses between ALS3 and HYR1. This was led by very talented postdoc at the time, Shakti Singh, who is a rising star as a young investigator at the Lankhos Institute. I don't see Shakti here, but I'm hoping he is listening to the talk. And you can see that we've had approximately 140 vaccine formulations that we basically screened. The screening was reliant on 2 things, antibody production as well as T-cell induction. So either by doing 2 doses or 3 doses of vaccine. So basically the regimen is to prime the mouse, wait for 21 days, vaccinate them with another dose, and you either vaccinate again with a 2nd dose, a booster, or you basically take them after 1 boost. And you bleed them, you do ELISA and figure out how much antibody titers you have against both antigens, or you sacrifice the mice after 34 days or 35 days, collect their spleens, and basically do test their splenocytes by fluorospot to determine basically a Th1 interferon gamma producing, Th2 IL-4 producing, or Th17 IL-17 producing T cells. And you can see here in alum that, as expected, alum is a very good Th2 inducer with a very good antibody titers. Mainly, we have more titers, as you can see here, with HYR1 compared to ALS3. These are the different doses that we've tested. Every time we mention 10 first, it basically refers to ALS3. The second one refers to HYR1. So this one has ALS3 antibodies, no HYR1. This is 10 microgram. This is 3 microgram HYR1. And you can see 3 microgram can produce higher antibody titers. And if you move into MF59, which is the adjuvant used in the flu vaccine, you basically get more or less the same antibody titers, but in this case, both antigens basically were highly immunogenic, so you can produce anti-ALS3 and anti-HYR1 equivalently antibody titers. And if you look at CAF01, you get antibody titers, but not to the same extent, lesser amount, but seems to be also more bias towards HYR1 antibodies, and you go look at GCP, stands for glucan chitin particles, you basically get more of anti-ALS3 antibodies compared to HYR1 antibodies. Again, very high antibody titers. And BDX100 produces more or less the same style as CAV01. How about T cells? So Alan, I want to say to our surprise, because I thought that it might actually produce some Th2, but it doesn't seem like it produces much, at least not with the parameters that we set. And I think this is where the problem, with the first generation NDV3A. You look at MF59, you get a very high Th2 immune response in T cells against both ALS3 and HYR1. And CAV01, you get a much balanced immune response between Th1, Th2, and Th17. GCP was more biased towards Th1 and Th17. And finally, BDX1 is biased towards Th1. So we focused on two antigens, two adjuvants, CAV01 and BDX1. And we've tested at least 2 doses, if not more, 30, 30, 30, and 10, 30. So the regimen for Candida albicans, we had to basically vaccinate 3 times, a prime and a 2 booster. If you do less than that, then you don't see much of a protection. So when you do this with the CAV01, as you can see here, different formulations of the vaccine, you get pretty much the 40 to 50% overall survival, long-term survival after day 21. If you look at BDX100, you get protection as well, maybe a little bit less than what we see with CAV01, but definitely a very significant protection in all the formulations that we've tested. You look at Candida auris, you need only 1 booster. So you prime, you wait for 21 days, and you boost, and then you infect after 2 weeks, after 2 weeks after the boost. So at day 35. And again, CAV01, all formulations seems to be actually highly protective. BDX1, still protective, but also, again, compared to the CAV01, those curves look slightly better. There is more clinical experience with CAV01. We felt also manufacturing-wise, it was CAV01 at the time when we tested it, it was in the hands of the Statin Serum Institute in Denmark. And then since we finished the studies, it was moved to a company called Croda. And Croda is basically producing it for clinical trials, and it has been used for at least 9 or 10 clinical trials so far. So we believe at some point, definitely a vaccine formulation is going to be approved with a CAV01. So when you look at the effect on CFU as a secondary endpoint, you look at the mice weight in the vaccinated mice, you get less weight loss compared to the placebo, as you can see here. And also you have approximately 1.5 log reduction in kidney CFUs, which is the primary target organ. The vaccine was also effective in reducing the vaginal infection, both different formulations at 10, 10, and 30, 10. And it can actually reduce it by almost sterilization at 3 logs, which is really quite impressive, I have to say. This is in Candida auris. The same thing that can be said about the Candida auris. Less weight reduction in the vaccinated compared to the placebo. And also it reduced the tissue fungal burden in kidneys, hearts, and brains. These are the target organs as well. We've also determined what is the mechanism of protection. So what we did is we vaccinated mice, and then we bled them. We took the serum, and we used that serum to basically treat naive mice that have been infected with Candida albicans or Candida auris. And you can see here that the antibodies, as shown here in red, they survive longer, but it's not really significant, but it's trending towards the right thing. If you look at mice that have been depleted of T cells by antibodies, and I'm just going to, this is a little bit complicated. If you look at the solid lines, the black one is the placebo. The red one is the mice that have been vaccinated. So clearly there's a difference between this and this. If you deplete T cells, that difference basically goes away. So the requirement of T cells is an absolute must in this vaccine. And the same thing can be said for Candida auris as well. Now, how long is the duration of the vaccine? So we did basically both the 2 doses versus 3 doses. And you can see that mice have been followed for 9 months, 270 days. And you see that there is basically a very steady antibody response against both ALS3 and HYR1 in here. And of course, the 2 booster was more impressive than the single booster, as you would expect. And the same thing for interferon, so for T cells. The only thing in T cells, you get basically a spike around day 28, and it starts going down. It doesn't really go down to the base level. You still have some T cells all the way after after 270 days. And if you look at mice that have been vaccinated and infected 4 months after the vaccine, you can see that there is basically a protection in the vaccine. I have to emphasize that patients who are going to get this vaccine, especially disseminated candida, they don't need to be protected for 9 months. You know, you think about it, these are patients who are going into a certain procedure. They're not going to stay in the hospital for 9 months, hopefully. So we're talking about a small period of time, probably no more than maximum, I would say, you know, most critical would be probably a month to a month and a half. This is basically what we're looking at. Of course, in terms of RBVC, maybe perhaps the vaccine has to be given on an annual basis. We also looked at whether the vaccine is effective with anti-fungal agents, and the reason for it is you vaccinate people, and if you have an outbreak, you have to treat anyway. And you can see here, excuse me, patients who basically get the vaccine, or sorry, mice that get vaccine, they are basically protected. Also, if you treat with sub-inhibitory concentrations of fluconazole, they are protected to the same thing. And this was basically used with sub-optimal doses because of 2 things. First of all, if you give an optimal dose, they basically protect mice completely, and you're not going to detect any synergy. So you have to go with something less than that. And if you basically vaccinate and treat at the same time, what you see is you get higher protection. It's not significant compared to the others, but you're definitely heading in the right direction. At least they are not antagonistic. This is both for Candida albicans and Candida auris, and both with azoles and echinocandin, which are really the major antifungal drugs used for Candida infection. So to summarize on future directions, this is a dual antigen vaccine, or what we call now in CAV01 VXV01, seems to basically induce a robust immune response, both in antibodies and T cells. The protection seems to actually work against both Candida albicans and Candida auris, and we're going to basically test against other species soon. CAV01 seems to be better, and all in all. And we know that the protection requires definitely a T cell immune response. Anybody might play a role, but not the prominent roles. T cells are really the prominent role. So where do we stand right now? We basically have generated MCBs from both producing cells, both ALS3 and HYR1. Then this is through using the preclinical service at NIAID. If you guys are not aware of it, just talk to me, and I can tell you and probably tell you who to talk to, at least for fungal infections. And also, we've just gotten, as Joe basically mentioned earlier, a broad agency announcement contract that is worth up to 40 million. We don't have the 40 million all at once. The base is basically, I just want to emphasize that. So we've gotten 3.6 million for the base for 2 years. And we're hoping that as we are generating more data, the options are basically mapped all the way up to 40 million to take us all the way to 2 phase 1s, one in adults and one in seniors. So let me switch gears and talk about mucormycosis. So I might have to skip the toxins, but at least I should get through basically the antibodies. So mucormycosis is a very lethal fungal infection. It's a major, major problem. If any clinician here probably will tell you how dreadful this infection is, and it is really a real emergency if somebody has it. It's rare, and probably I would say it's uncommon. It's not as rare as it used to be. I keep hearing about infections all the time now. It usually happens in immunosuppressed patients, and the mortality rate can be, overall mortality rate is 50%, can be as high as 100% depending on the host and what is the manifestation of the disease. And without going into too many details, this disease became a major issue during COVID because a lot of patients in India in particular got infected with mucormycosis because of the long period and high dose of steroid treatment to stem basically the hyper-inflammatory immune response to COVID. But they succumbed to infection with mucormycosis almost always in the diabetic setting. So those patients were diabetic to start with. So there are three major manifestations: rhino-orbital disease, as you can see here. This is a patient that was treated by Jack Edwards a long time ago. And you can see that when it happens in the sinus, it really necessitates surgery to remove all infected foci. And that means usually the removal of one eye, if not both. Pulmonary infection happens in neutropenic leukemic patients most of the time. This is mainly in diabetics and rhino-orbital disease. And this is cutaneous, which happened basically in severe trauma, such as natural disasters, the Joplin tornado. There was a cluster of 34 cases of infections, and in foot soldiers who were in Afghanistan, blown up by IEDs as well. So it's highly injury invasive. It likes to go after blood vessels, and it's very necrotic. So we've identified a cell surface protein called cut-itch protein, without going into details on how we did this. And the bottom line is, if you basically downregulate the expression of cut-itch, you prevent the organism from invading mammalian cells, both lung epithelial cells and endothelial cells. And also, it reduces the ability of the organism to cause damage to those mammalian cells. And if you take basically that attenuated expression stream, you can see that there is attenuation of virulence as well, as you can see here. Okay. And George and Abdullah were responsible for identifying the receptors, mammalian receptors, for cut-itch. So, and we figured out why diabetics, in particular, they are actually prone to rhino-orbital disease versus neutropenic in neutropenic leukemic being prone for pulmonary infection. So basically what happens in nasal epithelial cells, GRP78, which is glucose-regulated protein, gets upregulated in the settings of diabetics, meaning in hyperglycemia, in the presence of elevated available serum iron, in ketone bodies, especially diabetic ketoacidosis, and it acts as a receptor for cut-itch. So if you're inhaling, if you are in ketoacidosis and you are constantly inhaling, there is exposure. Rhizospus and mucorales are all over the place. And if it happens to be that you're inhaled mucorales and they find the receptor, they basically lodge themselves on the receptor, start proliferating and causing infection. You know, and that's why you basically have the sinus infection. You know, whereas GRP78 is not really well expressed in lung epithelial cells. And if you look, if you look at the other receptor, which was responsible, Abdullah basically found that EGFR receptor is basically responsible for lung epithelial cells and integrin as well. So what we believe is happening, that the rhizospus binds to integrin, the integrin activates EGFR, and that basically induces endocytosis and infection as well. So with the help of Vinnie Bruno at University of Maryland, we were able to sequence approximately 40-something mucorales. All of them are clinical isolates. All of them, they contain cut-itch proteins. And I should say that cut-itch proteins are really unique to mucorales fungi. They are not present anywhere else. They're not present in mammalian cells as well. And you can see that it can be present in anywhere between 1 copy to 7 copies in different organisms. And it's present in Rhizospus more than any other mucorales. And again, Rhizospus is the major cause of mucormycosis. Probably 70% of all causes of mucormycosis is related to cut-itch. Not only they are present, but they are one of the highly expressed antigens as well, both on spores and hyphae. And spores are the initial way that the organism is inhaled. Hyphae is the destructive invading form of the organism. So the idea here is to basically try to do a similar approach to the vaccine, but in this case, we try to raise antibody to block the ability of cut itch to bind to its receptor. And hopefully by doing so, we'll prevent basically early steps of infection. So with Mike's help, we were able to basically identify a peptide that is basically residing here in the binding facet. This is cut itch in yellow. This is GRP78. And you can see here that you can identify this binding facet, and identified approximately 16 amino acid peptide. We've raised antibodies against it, and we were able to isolate a mouse monoclonal antibody called C2, which was actually highly protective by itself. This is an animal model whereby we infect diabetic mice. We wait for 42 hours before we treat them to mimic severe disease, because it's really not easy to diagnose the disease to start with. And also, we wanted to see whether there is synergy between the antibody and antifungal drug, antifungal therapy. So you can see here that the antibody by itself is protective approximately 50%. If you actually do it 24 hours, it goes all the way to 70, 80%. This is with posaconazole. It wasn't really protective. If you combine them together, we get 100% protection. These mice are looking healthy. And we've done the same thing with liposomal amphotericin B. So both first-line therapy and salvage therapy seems to actually work synergistically with the antibody. So with these results, we started basically thinking about humanizing the antibody. So EU basically was responsible for testing several clones, humanized clones that we've done commercially. And you can see that those clones very nicely recognize yeast cells that are expressing cut itch protein, but they don't bind to any empty plasmid, right? So it's really nicely binding. VX-03 is a little bit dirty compared to the other two. And we can see that also all these clones bind very well to spores of Mucorales, different Mucorales. These are basically either treated with isotype matching control antibody. These are all IgG1s, or basically secondary only. And these are the ones that have been treated with the different clones of humanized antibodies. And it binds to Rhizopus oryzae, Cunninghamella, Mucorcercinoides, rhizomucorlecthymia. This represents approximately 95% of all causes of mucormycosis. Every now and then you'll get one of the exotic Mucorales that causes infection. Excuse me. So the humanized antibody maintained two important features. One is they were opsonophagocytic of the mouse monoclonal antibody. We've produced that in a publication in Science Advances 2019. And you can see that the VX-01 was equally opsonophagocytic. It enhances the ability of neutrophils to kill the organism in vitro. Also, the antibody is protective against invasion. This is, excuse me, for HUVEC cells. And if you look at damage, these are basically HUVEC cells that have been just sitting by themselves. This is treated with the antibody, humanized antibody by itself. You notice that there is no damage whatsoever, which is really good. It's a feature, a safety feature. And if you look at those that have been treated with Rhizopus delamar, you have actually a very good amount of damage caused by Rhizopus delamar, but when you put the antibody, you protect against the damage. Clearly, there are other factors as well that contribute to the damage, and we know what they are. They are toxins as well. And not only the antibody protected against damage, but also it protected against what we call vascular leak. This is a very important feature in fungal infections in general, but also in Rhizopus as well. In an assay that is reliant on fluorescent dextran, you basically can put confluent endothelial cells on a membrane, and then you put that fluorescent dextran in the presence of the antibody. You can see you have less fluorescent dextran detected in the lower chamber. In other words, it protects against basically vascular induced by Rispus delamar. So how does it do in animal model? So we've done, again, the severe animal model because we're interested in determining the antibody with antifungal. This will never be a standalone therapy. This is always going to be given with antifungals. So in an animal model where we infect mice, we treat them 48 hours post-infection, you can see that basically the antibody by itself, VX01, was protective, not so much by itself. If you look at ambisome, also wasn't protective. If you combine them together, you get 70% protection. And this is following 21 days post-infection. These mice look really happy and healthy at the end of the experiment. Not only it protects in survival, but also it lowers the tissue fungal burden, both in primary and secondary target organs. These mice are infected intratracheally, so the lungs are the primary, brain is the secondary because they disseminate to the brain. And again, the combination, you can see how synergistic it is compared to the monotherapy, compared to the placebo in both organs. We've done the same thing with posaconazole, by the way, and we get exactly the same thing. The mechanism of protection in vivo is really reliant on prevention of invasion. If you look at vascular endothelial cell growth factor as a marker for angioinvasion, in the absence of the VX01 and in the presence of an isotype matching IgG1, you get a lot of VEGF expression in lung tissues. And if you treat those mice with VX01, you get less, basically, of expression of the VEGF. And you can quantify it here, as you can see. We haven't detected any, basically, because we've said that these antibodies can be opsonophagocytic, so you would expect that macrophages probably, this is in neutropenic mice, by the way, macrophages can actually, tissue macrophages, that is, can basically show enhancement in the lungs, but that's not what we see, and we believe this is basically because of the prevention of invasion and invasion of the tissue parenchyma. We've also used those antibodies as prophylaxis. You can imagine certain patient population, especially those who are at a high risk of producing mucormycosis, bone marrow transplant, allogeneic bone marrow transplant in particular, you could probably put those patients on antibodies even before infection, and you could treat the treatment probably once every month or every 2 months based on the half-life of the antibodies. So VX01 has a half-life in mice approximately 5 days, which is expected to translate to probably 3 to 4 weeks in humans. And you can see here, if you infect mice after treating, 3 days, you actually protect them very well against the infection with Rispus delamar as well. One of the things that I have been asked repeatedly on what is the possibility of resistance against this antibody. I think it's safe to say that it is really very low. And the reason for it is VX01 targets gut itch, and it seems that there is no effect on the growth or germination of Rispus. It just binds to it without really killing the organism. So I don't think the organism is going to develop resistance to it. So to test this possibility, what we did is, actually when I say we, meaning EU, he took basically Rispus and passaged Rispus in the presence of VX01 for 20 passages. And then he took that organism that had been trained and used it to infect mice and compare it to mice that have been infected with Rispus that haven't seen the VX-01. And then we basically treated with either VX1 or the isotype matching control antibody. And you can see these are the two strains that we've used, the trained one and untrained one. They cause more or less the same mortality. And the one that have been passaged is in blue. The one that hasn't been passaged is in purple. And more or less they are superimposed. So we don't see that developed resistance. And also, if you look at the one that has been passaged, the antibody actually still binds to its target very well as well. So where do we stand with VX01? We've done GLP tox tissue cross-reactivity studies in human tissues, and seems to be there is no concern. We have done all manufacturing. We know for sure that we can actually produce it from CHO cells, approximately 3 grams per liter. We've done scaling up to 50 liter and then 200 liter as well. All methods have been developed. We have a stability study going till 12 months, no issues so far. We've done our pre-IND meeting in September. We've had a very good interaction with the FDA. They are willing to work with us on the phase 2 clinical design. It's going to be competitive, but they are going to allow us to use a hybrid system in the placebo. In other words, we can probably use historical data in the placebo as long as we enroll some patients as well. And also they are happy to have an adaptive clinical trial as well. Currently, we're doing GLP tox. We're done with all the dosing. We've done 5 doses in rats. No issues whatsoever. It's really very safe so far. We've done basically necropsy on the main study. We're doing the necropsy hopefully in a week or 2 on the 5 weeks. Maybe we've already done it. I'm just losing track. But so far, so good. Doesn't seem like this is going to be an issue. Antibodies are usually very safe, to be honest. So it's, and also we don't have any target in humans to it anyway. So we've done a BAA application in 2025. We haven't heard about it yet. Hopefully, if this is approved, we'll be able to take it all the way to another 2 phase 1 clinical trials. And then we would basically have to team up with another company to do the pivotal phase 2. There is no phase 3 for this drug or this antibody. I'm just going to skip because I know I'm running out of time. The toxins, and this could be probably for another day. So let me just go here quickly. Basically what it is, we have a toxin that acts like ricin. The toxin seems to be really important for necrosis. A lot of work was done by a talented postdoc at my lab. He's an associate professor at the University of Sharjah right now, Sameh, and Clara was a postdoc. And we've done work with Mark Swidergal as well. We know that the toxin specifically targets neutrophils in particular, to some extent less lymphocytes as well. And let me just finish by thanking people. I mean, this is Jack Edwards, as many of you know, who recruited me as a postdoc, and he was an absolutely fantastic mentor to myself and many others in the Division of Infectious Disease. He's the one who came up with the idea of using vaccine against Candida. And I give him all the credit for basically taking that vaccine all the way through NDV3A and Novodyne Therapeutics Inc. And hopefully in his legacy we continue, and hopefully we can produce a vaccine against Candida. There are many people on the list. I don't have the time to mention everybody. I mentioned most of them during the talk, and I am really indebted to all your help and all your teaching. You know, and I've learned from you guys a tremendous amount of knowledge, not only just scientifically, but personally as well. And for that, I thank you, and thank you all for listening as well. Thank you so much.
I just want to, it's not a question, it's just a comment. As someone who is a 4th-year student who was working on candidiasis and went into genetics instead, but with Jack Edwards when he was still a fellow, this is a over 50-year development, because that's how old I am in that career. This is over a 50-year development, and it's a wonderful example of course, the people involved, Ashraf, Scott, Mike Gaiman, et cetera, Jack Edwards, who has already been extremely well acknowledged. But it's just an example of what the Lundquist can do. and we have other examples, et cetera, but this is just a superb one, and it really is an example of continued persistence. Even, I mean, that's really good, fantastic how yet one company fails and you go on with another one. It's really great. No, I'm very serious about that. I am very serious about that.
And I thank you, Jerry, for saying that, and I fully agree with you. I mean, it's really a wonderful place to be here. I learned a lot from a lot of people here. It's a very, very nice and collaborative environment. Boy, it was probably really an awful talk if no questions. Come on, guys. Mark.
Hey, Ashraf, that was wonderful. And also, thanks for the shout out. I was just wondering with your COT-H3 vaccination, because you said it can enhance phagocytosis, but then you showed the macrophage data. Did you look earlier time point? Because it could be because you also get a reduction in fungal burden in the lung that correlates to the immune cell population. So if you would look earlier, and then specifically for in the BAL, so if you think that they're not going to get invaded, so maybe macrophages will like take it up of the alveolar spaces.
Yeah, I mean, this is a very good comment, Mark, and I think you do have a point. I don't think we've done many time points. I think this was a one point that EU basically done in response to a critique when we basically wanted to publish this thing. But this is something maybe perhaps we should go back and look at it just to see whether there is any involvement of macrophages and the opsonophagocytosis in the neutropenic model. I always thought, even in neutropenic mice, and there is evidence also in neutropenic patients that you still have basically tissue macrophages involved. And one way or the other, they probably have a role. Thank you.
Hey, Ash, it's Mary, and I'm online. Is it okay to ask a question?
For which one?
The Mucor one.
Yeah, so the plan is, if the BAA gets funded, then the plan is to take it into 2 phase 1 clinical trials. One is going to be in both of them are going to be healthy volunteers. One is in adults, one in seniors. But more importantly is the phase 2 pivotal study. What is it going to look like? We've tried to convince the FDA that we want to go and do open label single arm, but the FDA didn't agree to this. I should say that when we did our pre-IND meeting with the candidate vaccine, we've gotten a written response, no invitation for a meeting. When we've done the pre-IND for the monoclonal antibody against Mucor, they gave us a written response and they said, we're happy to meet with you face-to-face. So we took their invitation and we met with them on September 15th. So the FDA was extremely engaging and very helpful, I have to say. We've had a very, very pleasant meeting with them. And they said, you can't do historical data because this has to be a superiority study. So you're going to have to treat the humans with antifungals, regardless of what the antifungal is, ambisome or salvage therapy, posaconazole or isavuconazole, and the antibody with it. And they said what you do is you do a comparative clinical trial. However, we agree that this is really tough to do and it will take a long time to power it. So we are okay with you supplementing the placebo arm, the one which is going to be treated with antifungals alone, with historical data from the Fungoscope, which is a well-defined database, which, as you know, it was used in the VITAL study for approval of isavuconazole. So right now, we are consulting with Cytal, which is a company that does a lot of basically biostatistical analysis for clinical trials for Basilia and others. And we're in the process of trying to design this study. And we're getting a lot of help, I should say, from Youngju Park. So I think probably the goal here, Scott, is to basically take the well-defined mortality rate in the neutropenic leukemic patients, which was published in the VITAL study, usually hovers around 40% at day 42. Try to improve it by 20%, probably to 32% mortality at day 42. And it seems that they agree with us, the FDA. And they said this is good enough. So if we do this, I think we're gonna probably need 120 patients total, 60 in each arm. I'm hoping that they will allow us to do 1 to 3 in the placebo. So we're basically talking about an 80 patient study, 60 in the test, 20 in the placebo. The other thing is we said that we're gonna try to enroll patients in the study before they get basically definitive diagnosis, just on suspicion. And they agreed with this as long as definitive diagnosis can be done within 7 days. And they said, and we said the data basically that those patients haven't been diagnosed with mucormycosis can be used for safety. As you know, safety is an issue because we're not going to be able to give it to many people. And they agreed with that as well. So I think, you know, if we get an 80 patient study, you're probably talking about $10 million study. So it's not unusual to do this in a pivotal study. I mean, we've done market analysis on this antibody. It's not $1 billion. It could be $200 to $300 million only in the US and in Europe. It would be a good, you know, generating revenues for companies and for the institute if it happens. So, Abdullah.
How about the dose for the patients? What's
Yeah,
the dose?
so it's a good question. So the dose for animal models is approximately 30 microgram. If you extrapolate this to humans, it would be probably approximately 1.5 milligram per kilogram. So we're talking about, you know, this is not a huge dose. You could go higher than that. So we basically proposed to do the phase 1 clinical trial is at the dose of 30 microgram extrapolated to human dose and then 10 times fold higher in safety. And we can just decide on what needs to be done, you know, in phase 2 based on phase 1. Well, thank you so much, guys. Appreciate it. Thank you.
It's almost over. You're going to be a hard act.
Well, let's hope that we're able to get it to both of them to clinical trials. That would be really good.
This is sort of a naive question, so I wouldn't ask it. But in terms